Welcome, students. I'm back again with another very important topic, that is the chromosomal anomalies. Now, this is a very important topic in your genetics portion of zoology in the class 11th. Now, what are chromosomal anomalies? You all know that in case of humans, there are 23 pairs of chromosomes. Okay, so there are how many pairs of chromosomes? There are 23 pairs of chromosomes. So we shall be very brief with this, and later on we will begin to discuss the different types of the chromosomal anomalies in humans. If the number of chromosomes is different from this 23 pairs, that's different from 46 chromosomes, we say that there is some error in the number of the chromosomes, and that we define as the chromosomal anomaly. For example, we say that there are 47 chromosomes instead of 46. That's there's one extra chromosome in this condition, or there are only 45 chromosomes, that is there's one chromosome less than the normal number of chromosomes. This condition is defined as the chromosomal anomaly or chromosomal disorder. So after that, you will see, for example, there are different types of the chromosomal anomalies, but I will be very much specific over here with respect to the need of your syllabus and curriculum. If the chromosome number is diploid, normal diploid chromosome number, if the chromosome number is normal diploid, it is represented as 2n. It's the normal chromosome number, it's represented as 2n. If the condition is like this, it is 2n plus 1. For example, 2n number in case of human is 46 plus 1, means there is one extra chromosome. This very condition is defined as the trisomy. Okay, then if the condition is like this, 2n minus 1, the condition is 46 minus 1, it is described as monosomy. The conditions can be different from this also. It can be 2n minus 1 or 2n plus 2, but there are different cases. In your what we call as 12th class, you are asked only about monosomy and trisomy cases. Basically, we are dealing with 12th class zoology of the genetics portion. You are asked only about trisomy and monosomy. So we say that any abnormality in the number of the chromosomes is defined as the chromosomal anomaly, and two main cases of the chromosomal anomaly are trisomy and monosomy. If the chromosome number is 2n plus 1, it is trisomy. If it is 2n minus 1, it is monosomy. Then sometimes there's a condition which we call as the polyploidy. Now, what is the case of polyploidy? When you study the mitosis or meiosis in general, when you study the uh, cell division, you see at the telophase, after the telophase, there are prophase, metaphase, anaphase, followed by telophase. When the telophase is over, after that, there occurs cytokinesis. That is, the cells will now separate from one another. For example, this is a single cell. It will divide into two cells. So now the cytokinase occurs, so that chromosomes get distributed between these two cells. If at the end of the cell division the cytokinesis doesn't occur, so we say, again I repeat, if at the end of the cell division cytokinesis will not occur, it means all these chromosomes will get accumulated in the same cell, and it will increase the number of chromosomes manifold, and that very condition is known as the polyploidy condition. Okay, so you have to remember two main conditions of the chromosomal anomalies, that is the trisomy, that's 2n plus 1, in which the chromosome number would be in case of humans 47, and monosomy, that is 2n minus 1, in which the chromosome number would be 45 instead of normal 46. Let's take the first condition of chromosomal anomalies, which is in your syllabus, that is the condition of Klenfelters syndrome. Klenfelters syndrome. As from the title, it is obvious that this condition was first reported by this disorder was first reported by Klenfelter. This condition was first reported by Klenfelter. That is why this condition is known as the Klenfelters syndrome. So this was first reported by Klenfelter. Now what happens in this very condition? You see the normal chromosome number in case of humans is 46, but in Klenfelter syndrome the karyotype in Klenfelters syndrome. Karyotype means the chromosome type and number. Klenfelter syndrome it is like this: 44A means 44 autosomes plus XXY. So if it's a male, then the chromosomes should have been XY, but there is one extra X chromosome, so we define this as the trisomy of X chromosome. It is same as the condition of 2n plus 1 because there are 47 chromosomes instead of 46. So there are 44 autosomes plus 3 sex chromosomes. 44 plus 3 is equal to 47. So it's a case of trisomy, but it is a case of trisomy of sex chromosome. One extra X chromosome is present over here. That's why we define Klenfelter syndrome as a case of trisomy of X chromosome. So it's clear that how we define the Klenfelter syndrome is that it is a sex chromosomal anomaly and it is a trisomy of X chromosome. The chromosomal formula of the person suffering from the Klenfelter syndrome would be 44A plus XXY. Is it okay? Now we should move on towards the genetics of this very syndrome, genetics of Klenfelter's syndrome, in which we will try to understand as to how these individuals which are suffering from the Klenfelter syndrome are conceived. So our title would be now genetics of Klenfelter's syndrome. Genetics of Klenfelter's syndrome. Again, I will write over here that the karyotype of the individual suffering from Klenfelter syndrome would be 44A plus XXY. Is it okay? Now, what is the genetics of this very syndrome? It means how these individuals are conceived. You have to understand over here that if a normal what we call as normal sperm means normal sperm has chromosome number 22A plus what we call as Y. This is androsperm. If a normal androsperm, you might have understood earlier when we are dealing with the sex determination in humans, that in humans two types of spermatozoa are formed, one with the chromosomal formula 22A plus X, that's known as gymnosperm, and another with the chromosomal formula 22A plus Y, which is known as androsperm. When this androsperm fertilizes an abnormal ovum which is 22A plus XX, and there's no ovum like this unless it is abnormal. The ovum should have been like this. It should have been 22A plus X, because during meiosis the autosomes as well as the sex chromosomes get separated from their homologous chromosome. But over here you have seen a condition whereas the XX pair has not separated. The condition should have been like this, that this X chromosome should have been separated from this X chromosome, so that the chromosomal formula in the ovum should have been 22A plus X. But over here what has happened, the disjunction has not happened. Disjunction means separation of XX pair has not occurred. There we say that non-disjunction means non-separation of XX pair during meiosis. So when meiosis occurred, there should have been the separation of XX pair, which we call as disjunction, but there has been non-separation of XX pair due to some error in the meiosis. We call that as non-disjunction. So by the non-disjunction of XX pair, an abnormal ovum, that's 22 plus XXY, has been formed. When this abnormal ovum is fertilized by normal androsperm, the offspring would have 22 plus 22, that's 44A, means 44 autosomes, plus this X and this X, XX, and this Y. This is the karyotype of the individual suffering from Klenfelters syndrome. So this is the first condition. You see, this is the first condition in which you have witnessed that how by fertilizing an abnormal ovum having 22 autosomes plus XXY pair of sex chromosomes, when it is fertilized by normal androsperm, how this Klenfelter syndrome individual has been conceived. Now there is also another probability in which you will see Klenfelter syndrome individuals can be conceived. And that very condition is like this. It is when an abnormal sperm with the chromosomal formula of 22A plus XY fertilize a normal ovum with the chromosomal formula of 22A plus X. So this is normal ovum having one X chromosome and 22 autosomes, but this abnormal ovum in which what has happened, non-disjunction, in which non-disjunction of XY pair has occurred. Non-disjunction of XY pair has occurred. The normal procedure is that this X chromosome should be separated from Y chromosome. That's known as disjunction. But there is non-separation. It is known as non-disjunction. So this is an error in meiosis has happened, due to which an abnormal sperm with 22 autosomes plus XY pair of chromosomes has occurred, and it fertilizes the ovum. So the resultant offspring would have 44 autosomes plus this X, and this Y. So this is a way the Klenfelter syndrome individual can be formed in the second case. So there are two conditions how the Klenfelter syndrome individuals are conceived. One is the condition in which the ovum is abnormal, in which XX non-disjunction has occurred, as we have studied in the first case. The second condition is in which the sperm is abnormal, in which XY pair of chromosomes have failed to separate from one another during the meiosis. Now, by whatever way from these two very cases the Klenfelter syndrome individual has got conceived, we will now understand the disorders in Klenfelter's syndrome individuals. First thing is that their karyotype means the chromosome number and type, is 44A plus XXY. So it's a sex chromosomal anomaly. It's a case of trisomy of X chromosome. Okay. Now, first thing is that they are males, but they are sterile males with reduced testicles. So they have reduced testes or testicles, so they are sterile males. They are not able to produce offspring. Second thing is that they have sparse, means very less dense, sparse hair on face. The beard, you see, it will be very thin in these very individuals. And then they have long limbs, particularly legs, with abnormal knees, which we call as knock knees. Okay. Then they have low IQ, low intelligence coefficient, so they will not be having normal intelligence. Another thing is that they would be suffering from gynecomastia. It's an abnormal condition of development of breastes in males. Breast is basically developed in case of females. If they develop in case of males, it's an abnormal condition and it is known as gynecomastia. So these are the different types of the disorders which occur in the Klenfelter syndrome individuals. Now we will move on towards the second type of the chromosomal anomaly, sex chromosomal anomaly, that's known as the Turner's syndrome. Let's move on towards the next type of the disorder, which we call as the Turner's syndrome. Turner's syndrome. No need to write over here that this disorder was first of all reported by Turner, that's why it is known as Turner's syndrome. Now these individuals have a karyotype means chromosome type and number. Karyotype have 44, karyotype of these individuals is 44 plus X0. So it's obviously a case of 2n-1, that's one chromosome less than the normal count of 46, that's 46 minus 1, that is 45. So this very case, 2n-1 case, is known as monosomy. It is a case of monosomy, it's a chromosomal anomaly, and it is a sex chromosomal anomaly. So we say it's a case of monosomy, it's a case of monosomy of X chromosome. The case should have been, if this might have been a normal female, the condition would have been 44A plus XX, but there's only one X chromosome, that is, it is 44A plus X0, that's why we say it is a case of monosomy of the X chromosome. Now let's move forward towards the genetics of this very type of the disorder, as to how these individuals are conceived. The first thing is, if a normal gynosperm means the individual having 22 autosomes plus X chromosome in its sperm, that's known as a gynosperm. Gynosperm has a chromosomal formula of 22A plus X. Fertilize an abnormal ovum having only 22 autosomes plus 0 X chromosomes, means no X chromosome. How it has happened? It has happened due to the XX non-disjunction. Over here the X and X should have been separated, but they do not got separated. As a result, they went in one ovum, and in another ovum there is left no X chromosome, there is 0, and this condition is 22A plus 0. If this sperm fertilize this ovum, the resultant offspring would be like this: 22 plus 22, that's 44A plus X and 0, X0. It is known as the Turner's syndrome karyotype. This is the Turner's syndrome. Is it okay? Clear? So this is the first case how a Turner syndrome individual can be conceived. Now let's move on towards the second case. How the Turner syndrome individual can be conceived. The second case we are taking over here. In the second case, we will deal with the abnormal sperm in which there are only 22 autosomes plus no X chromosome, that is 0. This abnormal sperm, if it fertilizes the normal ovum would have chromosomal makeup of 22A autosomes plus X. Is it okay? It's normal ovum. This sperm fertilizes this ovum, the resultant offspring would be 44A plus X0, and this individual would again be suffering from Turner's syndrome. So you have understood how the Turner syndrome individuals are conceived. Now let's move on towards understanding the disorders in these individuals, that is Turner syndrome individuals. The first thing is that they are sterile females, that they are not able to produce offsprings with karyotype of, they are sterile females with karyotype of 44A plus X0. Okay, this is the first thing. The second thing is that they undergo gonadal dysgenesis. That is, they have rudimentary and non-functional ovaries. The ovaries in these females are very rudimentary and they are non-functional. That is, we say they have underwent gonadal dysgenesis. Is it okay? The third point is that they have hearing and cardiovascular impairment. That is, they have problem in hearing and they can also suffer from some of their heart disease, which we call as cardiovascular ailments. Then there is another thing, that they have short stature. Their height is less, so we say they have short stature, and webbing of skin in neck region. It means that they develop, for example, this is the neck of such a female, they develop webs in the skin in the neck region. That's we call as the webbing of the skin in the neck region. So this is all about Klenfelter syndrome and the Turner's syndrome, which are both sex chromosomal anomalies.